The male reproductive system is a highly specialized biological assembly line. This continuous histological gradient is engineered to derive, modify, and propel haploid cells. Here, in this sagittal view, we can locate the testis suspended within the scrotum. The organ itself is subdivided internally by fibrous septa into roughly 250 distinct pyramidal compartments, known as testicular lobules. Looking closely at this cutaway diagram of a single lobule, we find it consists of two primary components: a sparse interstitial connective tissue and 1 to 4 highly convoluted loops called seminiferous tubules. This high magnification section isolates the interstitial tissue between tubules. These clumped polygonal cells are Leydig cells. Notice their eosinophilic cytoplasm packed with lipid droplets. These droplets provide precursors for steroid synthesis, allowing Leydig cells to synthesize testosterone when triggered by luteinizing hormone. This immunohistochemical stain highlights the germinal epithelium. The yellow fluorescence traces Sertoli cells, tall columnar cells extending to the lumen. Driven by follicle-stimulating hormone, they secrete androgen-binding protein to concentrate local testosterone and inhibin to regulate pituitary feedback. The testis's basal architecture functions as an endocrine-driven support engine. This strict hormonal and physical scaffolding is an absolute requirement to initiate gametogenesis. Elaborate tight junctions between adjacent Sertoli cells create an impermeable physical seal, dividing the tubule into isolated basal and adluminal microenvironments. This blood-testis barrier prevents patrolling lymphocytes from detecting developing haploid sperm. If compromised, leaked sperm antigens trigger immediate infertility-causing autoimmune attacks. Therefore, before a sperm cell undergoes further biological modification or mechanical propulsion, it must be anatomically sequestered from the host's own immune defenses. This diagram illustrates sperm derivation. Diploid spermatogonia in the basal compartment divide to produce primary spermatocytes. These migrate into the adluminal compartment through the tight junctions. Here they undergo meiosis, separating chromosomes to ultimately yield small, haploid spermatids near the central lumen. Look closely at the horizontal connections in this flowchart. The divisions from spermatogonia down to spermatids feature incomplete cytokinesis. The resulting cells remain tethered by intercellular cytoplasmic bridges, creating a clonal syncytium that allows them to share gene products across the entire connected network. In Panel B, we see the final phase, spermiogenesis. No further cell division occurs here. Instead, it is a purely morphological shift that converts the round, haploid spermatids into highly specialized spermatozoa. This transmission electron micrograph captures structural remodeling. A thin, Golgi-derived acrosomal cap, packed with hydrolytic enzymes, spreads over the condensed nucleus. As the cell elongates, DNA is tightly packed by protamines, a flagellum grows, and Sertoli cells phagocytose the shed cytoplasm. The testicular engine successfully manufactures a structurally complete gamete. However, this resulting spermatozoon is completely immotile, relying entirely on the tract's architecture for physical transport. The immotile sperm are pushed out of the seminiferous tubules by fluid pressure into a short transition zone called the straight tubules. These immediately empty into the rete testis, a complex labyrinth of interconnected channels housed safely within the dense fibrous tissue of the mediastinum. Sperm then enter the efferent ductules. This section reveals a distinct, scalloped lumen driven by an alternating epithelium. Short, non-ciliated cells absorb fluid to concentrate the sperm, while patches of taller, ciliated cells actively beat to sweep the still immotile gametes forward. This reliance on ciliary motion creates a vulnerability. In a condition called primary ciliary dyskinesia, particularly Carter-Jenner syndrome, a genetic mutation causes a complete absence of the dynin arms required for ciliary movement. For individuals with this condition, infertility is a direct consequence. The cilia in the efferent ductules cannot sweep the fluid forward, and the sperm's flagella, which share the exact same microtubule architecture, will fail to achieve motility later on. Without a functioning ciliary architecture to maintain the fluid current through this specific transit zone, the passive transport of immature sperm halts entirely. Leaving the efferent ductules, cells enter the highly coiled duct of the epididymis. This micrograph shows the epithelium shifting to a uniform pseudostratified columnar layer of tall principal cells. Their defining characteristic is the extremely long non-modal stereocilia projecting deeply into the lumen. Over the next 2 to 4 weeks in the epididymis, the sperm undergo critical biochemical maturation. The principal cells secrete lipids and glycoproteins, adding decapacitation factors to the sperm membrane to block premature fertilization, while simultaneously granting the gametes the competence for forward motility. Once mature, sperm move into the vas deferens. This cross-section reveals a drastic structural adaptation built solely for rapid transit. Notice the massive muscularis layer dominating the wall, organized into inner longitudinal, a remarkably thick middle circular, and outer longitudinal bands of smooth muscle. This overwhelming investment in smooth muscle is directly correlated to its function. During ejaculation, these 3 layers fire in aggressive peristaltic contractions, rapidly launching the sperm out of the scrotum and toward the urethra. The biological objective is no longer modification. The tract has definitively transitioned into a high-pressure mechanical transport tube. Just before the vas deferens merges into the ejaculatory duct, it meets the paired seminal vesicles. This anatomical diagram shows their position behind the bladder, generating up to 70. This expanded epithelium produces a viscid fluid containing fructose, the primary energy source for motile sperm. It also secretes prostaglandins, which act as local hormones to stimulate smooth muscle activity within the female reproductive tract, along with fibrinogen, which allows the semen to rapidly coagulate immediately following ejaculation. The labyrinthine architecture of the seminal vesicle is designed to manufacture and deliver the massive metabolic payload sperm required to survive outside the male tract. As the ejaculatory ducts descend, they enter the prostate gland. This diagram shows the prostate sitting directly inferior to the bladder, completely enveloping the prostatic urethra. Histologically, the prostate consists of 30 to 50 branched tubulo-acinar glands. As seen in this tissue section, these glandular elements are heavily embedded within a dense, pink-staining fibromuscular stroma. The epithelial cells lining these glands secrete a complex fluid containing prostate-specific antigen, or PSA. PSA is a serine protease, whose clinical and physiological role is to slowly liquefy the coagulated semen, allowing the controlled release of sperm. When viewing normal prostate tissue, you will frequently spot a corpus amylaceum, a lamellar, often calcified glycoprotein concretion residing inside the gland lumen. They increase in number with age, but carry no negative clinical significance. To map prostate pathology, we divide its anatomy into strict zones. The inner transition zone is the primary site for benign prostatic hyperplasia. Growth here exerts inward pressure, causing direct urethral compression. Conversely, the outermost peripheral zone is the origin site for malignant adenocarcinomas. In the prostate, spatial coordinates dictate the clinical outcome. The exact zone where cellular division goes awry determines whether pathology presents early as a benign mechanical blockage or grows silently as a malignant cancer. Zooming out, the reproductive tract operates as a single, regulated biological assembly line. The gradient begins with haploid cell derivation in the testis, passive transport to the epididymis for maturation, forceful muscled transit via the vas deferens, and terminates with accessory gland exocrine palo. True mastery of male reproductive physiology and its pathologies requires understanding this strict sequence. Every clinical failure, from autoimmune infertility to ciliary dyskinesia, can be traced back to a specific structural disruption along this continuous histological gradient.